Thank you very much, Anita. And thank you all for your attendance early this morning. And as Anita indicated, my interest is in encephalopathy of preterm birth. So we all know that preterm birth is still a leading cause of morbidity and mortality amongst the infants in our community. This means that we have a lot of children living with permanent disability, and we can see from this graph the lovely illustration that the earlier an infant is born, the more likely they are to need special educational needs. Maternal and fetal inflammation is not only a leading factor associated with precipitating preterm birth, but increasingly we understand that persisting inflammation in the infant in the neonatal period is also a very strong predictor of poor neurological outcome. Encephalopathy of preterm birth is the name that we give a constellation of neuropathological findings that we see in preterm-born infants. Because of the immense improvements in antenatal care and neonatal care that we're able to offer these infants, in no small part due to the immense activities by organisations such as all of you today. What we see is infants no longer present with cystic lesions, so really drastic changes in their brains, but more often they have diffuse lesions. And these are what we strongly associate with learning and behavioural disorders. So the hallmarks of encephalopathy of prematurity. If any of you have seen me present before, you'll know that my overarching focus has always been on microgliosis and how microglia drive brain injury. And collectively, we've always focused quite strongly in our community on changes in oligodendrocytes and how these lead to hypomyelination in these infants. But increasingly now, our focus has shifted to grey matter dysmaturation as the frank lesions in the white matter have become less common. So some studies that really started to illustrate for us collectively how important changes in the grey matter are is this study as an example from Gareth Ball, who is in King's College working with David Edwards, and both looking at cortical microstructure as an index from fractional anisotropy and mean diffusivity, we see when infants are scanned at term equivalent age, the infants that were born preterm have striking differences in their cortical microstructure across regions of the brain. And I suppose now, as we see these infants with diffuse white matter lesions as a collective grow up and go to school, and fortunately many of them still go to university, we've started to study their brains and how they are as young adults. And we do see that they persistently have reductions in global brain volumes, but quite interestingly, these effects are region-specific. And this paper by Chiara Nosardi and her team have actually postulated that there's a dysmaturation across various regions of the brain which do require further study, and that's where people such as myself come in by using animal models to try and tease apart some of the effects that that clinicians are seeing in their patient cohorts. So we model neuroinflammation-mediated brain injury. So as I mentioned, we know that exposure to inflammation is one of the leading factors that predicts poor outcomes, and poor outcomes being changes in white and grey matter. So some of you will have seen our animal model before. So between postnatal day 1 in the mouse, P0 being birth, so between P1 and P5 we give intraperitoneal injections of an inflammatory stimulus. So the stimulus itself really isn't important. The fact that it stimulates neuroinflammation, microglial activation and damage to oligodendrocytes is really what we're after here, to mimic the inflammatory stimulus seen in preterm-born infants. And importantly, our model leads to changes visualised with imaging paradigms, behavioural sequelae, and also changes in those characteristic effects in oligodendrocytes and myelination. So we started investigating the grey matter in our model, And one of our initial studies was done in collaboration with Anthony Vernon from the IOP at King's College. And we were able to show quite simply in a way, but importantly, that in our model, when we scanned the animals at postnatal day 60, that there was an overall reduction in grey matter volumes in these mice. These localised to specific areas of the brain, but although we can't expect to see everything line up with the preterm born infant, we were happy that it was suggestive that it's worth continuing to look at the grey matter. We also started looking at gene expression. So we had gross morphological changes, but at the gene expression level, when we look in the cortex of this model, and we've done these analyses both at postnatal day 5, when our inflammatory stimulus is still ongoing, and also at P10, which is term equivalent age in our mice. And what we see is the gene expression changes in this model really suggest that we have striking changes in the way that the neurons are communicating with one another, key signalling pathways and ligand receptor interactions that are altered. So some lovely work to expand on this was done by a collaborator and close friend of mine, Helen Stolp, and using the same paradigm, she did analysis in these young adult mice, and she was able to show where these animals had been exposed to an inflammatory stimulus to mimic what's happening in many of our preterm born infants. What we see, this dark bar here, is showing that the inflammatory exposed animals are more likely to have sections of their processes with low spine density, indicating that there really is quite a marked difference in the way that these neurons are formed. So interneurons. Interneurons have almost become the sexy new thing for many of us in the translational field. And of course, we thought that the logical next step in our model was to begin investigations into these cells. So interneurons are a very heterogeneous group of cells, both morphologically, biochemically, and with their electrophysiological properties. Briefly, these cells are born in the preoptic area and the medial and lateral ganglionic eminence, and they begin a very long tangential migration before they integrate into the cortical layers. Now, the reason that these cells are so interesting to those of us working with preterm born infants is that the proliferation, migration and integration of these cells is still occurring during the last trimester, which is when preterm birth typically occurs, and through infancy, making these cells quite vulnerable to the effects of preterm birth and their associated injuries. We also know from human post-mortem studies in neurodevelopmental and neuropsychiatric disorders and in preclinical studies in these disorders that changes in interneurons are implicated in processes such as autism spectrum disorders, epilepsy and ADHD. And as many of you are aware, these disorders are preterm born infants have an increased risk of these disorders. So interneurons can be characterised, as I mentioned, by their morphology, by their spike properties and their connectivity. The studies that I'll show today focus very much on the biochemical signature of these cells because they help us understand how they're affecting the signalling within the cortex. So I'll give you a brief rundown of the three main groups of interneurons. So parvalbumin-positive cells, which are the largest class of interneurons and present across all layers. Somatostatin-positive cells, which really are the first interneurons that migrate into the deep layers during development. Oh, and if I say here, we also have calreticulin down here as a subpopulation. And the third class is a very heterogeneous group. And although it can be characterised by its serotonin receptor expression, a lot of us in the field, we typically look at the subpopulations specifically, looking at VIP and also looking at reelin expression. So mice are incredibly useful for these types of studies for understanding these cells, and a great deal has been done to map out the milestones. of interneuron development. And I'd just like to highlight here, we can see that, and say this tangential migration bar, for example, in the orange, it continues for quite a long time and overlaps with the processes both of neurogenesis, so the birth of the interneurons, and the laminar allocation of these cells. And that's because there are different locations these cells are born, and they're also born at different times and migrate at different speeds and integrate at different stages during development. What I'd like to highlight, though, is this window here between postnatal day 1 and postnatal day 5 in the mouse, which of course is when we apply our injurious stimulus in our animal model, and it's the approximate developmental window in which most of the cohorts of preterm born infants I'll describe were delivered. So human studies. So the project I'm describing today sort of happened in 2 parallel parts. There were post-mortem studies of preterm born infants, and we went into this project knowing that there had been a study showing that in preterm born infants with white matter lesions, there was quite a striking reduction in the amount of GABAergic interneurons. This was compared to infants that were born preterm but had no lesions. The caveat being that these cases were quite severe, and there was quite marked cell death with oligodendrocyte loss and severe white matter injury. So the cohort that we studied, and I'd like to really highlight that this cohort is exists because of the very hard work done by Mary Rutherford at King's College and support from both Vina and Regina. So these are the patient characteristics for the cohort that we studied. I'd like to highlight that, again, all of these are preterm born infants, and they were categorised into infants with no white matter injury and non-cystic white matter injury. So these samples were analysed using a semi-stereological approach with multiple counting sites in the frontal cortex. And where possible, depending on the staining, and we needed to be really reliably happy with the consistency of the staining in these cohorts, which is always difficult in human studies. We were also able to do some neurite length analysis. So the first cell type of interest was somatostatin. So we saw no change in somatostatin positive cell number, and this is sort of a common theme we'll see across some of our data, but really striking changes in the morphology, so neurite length and branching. Calreticulin positive cells, there was a really marked decrease in the expression of these cells across the layers of the brain, and neuropeptide Y. This is another cell type we were able to get the morphological assessment done on simply because of the nature of the staining. And although, again, there was no change in the number, it was remarkable to see actually how different the morphology of these cells were. So as I mentioned, we paired, or in parallel, we were working on our animal model. We started doing our analysis in this model at postnatal day 5. And as I indicated before, this is during a period where we expect both to have migration, laminar allocation, and probably also some wiring of interneuron subpopulations occurring. So some really beautiful work done by Yoko in our lab. She was able to do some pretty exhaustive sectioning and immunohistochemistry to show that as we'd seen from our MRI data, it was possible to visualise the reduced cortical volumes in this model. And she did some work analysing the various layers and the subpopulations of interneurons, including calreticulin and reelin, shown here. But then we also moved on when we've done parvalbumin, somatostatin, NPY, and calbindin. And we can see that there are some changes that appear in some of our images, but when we did our analysis, what we saw quite interestingly is that were changes in reelin positive cells, so we had reductions in this subpopulation of interneurons, and quite a striking reduction in the calreticulin. Now this is only at P5, which is quite an early time point, and we also broke down our data by layers as well. because of the previous knowledge we had about interneuron, the location in a different lamina. And all of these two significant effects here were really driven by effects in the lower layers, which is probably not surprising given that interneurons migrate into the cortex in an outside-in manner during development. So expanding out to P10, neuropeptide Y wasn't changed early, continued to not be different. The effects in calreticulin normalised, indicating that we need to chase it up, but that maybe we just had a delayed migration of these cells. And calbindin, we had no change early and no change late. So if we start here with the somatostatin on this side here, we can see that there's no change at P5 or P10 for our somatostatin positive cells. But our palvarbermin positive interneurons, which to remind you is our largest population of interneurons, there was no change in the numbers at P5, but across both the upper layers, so these layers here and layers 5 and 6, we saw reductions in the numbers. Now, we're a little bit excited by this because we'd seen some really amazing presentations, I think it might have been at Euroglia, on perineuronal nets, I think in the weeks before this data came from our students. And perineuronal nets are an amazing additional way that interneurons modulate their activity and stabilise their integration into the cortical circuits. This is a specialised form of extracellular matrix protein, and when the palvarbermin positive interneuron is established, this extracellular matrix protein accumulates, and what it does is it prevents new axons from terminating on the cell bodies, and it also stabilises the receptor migration so that you have less flexibility in the responses of these cells. And to illustrate what these look like, this is quite a beautiful stain to do actually. So down the bottom here is the palvarbermin positive staining in isolation, and we can see here in the double labelling image, we can see these little red dots, and they're our palvarbermin positive interneurons. And then using a Wisteria floribunda agglutinin, which I don't say out loud very often, we just stick with WFA, we can see that as these cells, the palvarbermin positive cells, have matured, they accumulate this beautiful halo of perineuronal net, and this sort of becomes elaborate down the processes as well. What this allows us to do is to discriminate palvarbermin positive cells that have fully formed and quite elaborate perineuronal nets, such as this cell here, and then to identify palvarbermin positive cells that do not have a fully formed perineuronal net. Now, even in a control brain, there are always palvarbermin positive cells that do not have a perineuronal net, and this has been reported by others as well, but it's typically said that the vast majority of these cells will mature and establish their own net. So looking across time, it kind of validated for us the steps that had already been reported in interneuron development in the mouse. So at P10, our little red dots, we can see our palvarbermin positive cells are beginning to invade and integrate into the cortex. We have, at P35, we have a vast number of palvarbermin positive cells, and a number of these are lost as the brain further matures, but the cells begin to express these mature nets towards the age of P60. So because we had seen at our P10 time point that there'd been a reduction in the number of palvarbermin positive interneurons, the question then was, how does this affect the maturation of these cells? So we expanded our analysis to P40, and at this time point we saw that the numbers had normalised, and we no longer had a specific difference in the numbers of these cells. But when we analysed the number of palvarbermin positive interneurons and whether or not they expressed a mature, fully formed perineuronal net, we saw that there was a reduction in the amount of palvarbermin positive interneurons that had one of these fully formed nets, which An absence of the nets has been associated with increased cortical circuit excitability, and it's gotten to the point now where there are experiments being shown to say that you can transplant into neurons and actually modulate the cortical excitability, which I think means that these changes are likely to have important functional consequences for the brain that we now need to elaborate on. So in summary, both in our animal model and in our human post-mortem cases, we did see reductions in interneuron numbers, and in our animals we saw changes which would indicate changes in function, perhaps. I think what's important is in the human cases, of course, we don't have multiple time points, so we can't identify if some of these changes will have normalised, as we saw in our animal models. Now, just to finish, I'd like to highlight that while we were essentially going through the review process and getting our data published, that a couple of further works came out on interneurons. And I'd like to really highlight this one from Anna Penn's group in the University of Washington. And they also did some human studies and some animal model studies. The really important difference here is, though, that they looked at infants that were born at term as their controls, and then infants that were born preterm as comparisons. Now, they saw really striking differences in the number of somatostatin positive cells and also calbindin, which we didn't see either of these when we looked at our human cohorts, but again we were comparing preterm infants with and without white matter injury. They also set up a double hit model of encephalopathy of prematurity of EOP, bringing together maternal immune activation, so systemic administration of a low dose of lipopolysaccharide, sort of late in gestation. And this was to mimic what they see in extremely preterm born infants, where a vast majority of these infants, their mothers present with chorioamnionitis. And then to mimic issues with lung function in the first week of life, they also expose these animals to hypoxia. Now, I think it's quite interesting. I just want to orientate you here. So there's 4 groups. There's the controls, immune activation only, which I think is most relevant to our own model of P1 to P5 inflammation. And here is the hypoxia only, and then the combined model. Well, what I'd like to highlight is that there are some really interesting complexities to this data, which builds on what we've seen. Complexities such as the fact that maternal immune activation alone tends to increase the numbers of somatostatin-positive interneurons in the cortex, but the double hit, of course, has probably what we'd more expect to see reductions, but there are very specific and discrete effects of immune activation only that I think we sort of need to get a handle on in terms of what the relevance of the timing is and what this means for our infants. Because when we talk about preterm infants, of course, we have so many subpopulations of these, and I think if there's a future for targeting interneuron deficits, our understanding and comparison across these preclinical studies is really important. And this paper, which was also put out by Veena and Anna, they worked on this in a project by their time in Sweden. And so this is a preterm sheep model. So if you, the sheep gestation is 140 days gestational age. Now, sheep are born more developed than a human, so 100 days gestational age is about moderate preterm. This is a hypoxia ischemia model. They assessed multiple brain regions in this study, but limited themselves to somatostatin. Now, similar to what we had seen in our human cohort, there were no reductions in somatostatin positive number, but strikingly there were these beautiful reductions in the arborization of the cells, which is quite visible here in the image taken from their publication. Now, I think what all of this tells us is that it is undoubtedly true that interneurons are vulnerable to the types of insults that preterm-born infants are exposed to, whether that be very early inflammation, chronic late inflammation, or variances in oxygen concentrations. But the complexity in the vast numbers of interneurons as yet means that we don't have a clear understanding of what's really going on and how we can tunnel through this data to come out towards a therapy that we can ask some of you to apply in your infants. So we do need more studies, and I'm hoping in the next few years we can come back to you with those. I'd like to thank everyone who participated to these studies, and of course, if you have any exciting work, please keep us in mind for our Frontiers special edition that's coming up.
Thank you. Thank you very much. So the paper is open for discussion. I don't know if there are some. Maybe I start with one question. You pointed out in your animal model that you are looking also for long-term outcome. Did you do anything yet, or?
Do you mean in terms of behaviours?
Behaviour, yeah.
Yeah, so we already have a fairly well-characterised set of outcome measures in this animal model. So we know that there's social anxiety and cognitive deficits in this animal model.
Okay. Yeah. Mm-hmm. Yes?
Petra Hüppi, Geneva. Thank you, Bobbi, for very interesting new data. I was just wondering, I think it's relevant for what we do in care. You talked about some of the hormones, you've left out others. I think to recall that there was a recent paper of estrogen actually rescuing interneuron development to some extent.
Yes.
And I was wondering what your view was on these hormonal interactions. They could also involve oxytocin at that stage. What do you think it does to this particular population of neurons?
Yeah, and that was just a time issue. I think that's the Panda paper. I can't remember. It's the rabbit kit paper as well. And I agree with you. The hormones are definitely important. One of the key problems with preterm birth is the absence of the placentally derived hormones, and I think there's been a push to have the artificial placenta, and of course the failures of that over the years. That's been on the cards now for about 40 years. Tells us we don't understand the normal hormonal milieu well enough. But I think the estrogen paper was really valid, and I think it's a great approach. And it's just now about getting people on side, because I assume there are issues with who would fund these types of trials with something like estrogen or progesterone or allopregnanolone, all these hormones that are missing.
Yeah, I think we're not there yet, but I think your models have to, give us some more hints of what the cocktail would be.
Yeah, and I think once we understand in our model a bit more about exactly what's going on, it'll give us a handle on starting to do those neurotherapeutic trials.
Other questions? No?
Quick question, Bobbi. I'm wondering why we've missed them all these years and haven't taken notice of interneurons. But as for the percentage of brain cells and the importance, can you give us an idea of how many compared to neurons, astrocytes, are we dealing with?
The estimates range between 10 and 20% of cortical cells.
Okay.
So, yeah, we're talking about a lot, but I think, as for so many things in science, they fall in and out of favour. As someone who's on the microglial bandwagon, I think many of you probably remember the astrocyte bandwagon. So I think it's just about the tools and techniques as well that are available.
Okay. Any other questions? If not, I think we move on. Thank you very much, Bobbi.
Thank you.